Hello everyone, today we're going to start with our biochemistry course. Now, first of all, we will start with the difference between eukaryotic and prokaryotic cells. What are those? Let me write eukaryotic and prokaryotic cells. In eukaryotic cells, we have membrane-bound nucleus, membrane-bound nucleus. Why is this used for? It is used in order to protect the DNA, right? We need to protect the DNA as eukaryotic cells. And then the second function, of course, make selective import and export via nuclear membrane pores. So, as we will discuss in a minute, the nuclear pores are enclosed by a sphere, and we have selective import and export because we need to be this system as a cell to be not in equilibrium, because if it were in equilibrium we would be dead, right? Equilibrium means death in living organisms. So in order to maintain homeostasis, we need a selective membrane that keeps the some of the stuff out and takes some important materials inside of the DNA. All right, let me delete this and continue. Here we write selective import and export. All right. And then we have membrane enclosed organelles. What about those? Do we have any examples of those? Of course, for example, we have mitochondria. We have chloroplasts. And of course, lysosomes. We will discuss the functions and how are they positioned in the cell in a minute, so that we can easily understand how what is their task throughout the cell. However, in prokaryotic cells, what is the difference? The difference is that the nucleic acid, the genetic material, is not enclosed by any membrane, nor any organelle is enclosed by any membrane. No membrane, let me write. It is found in bacteria, in archaeobacteria, etc. Now, it is important to know how the cells are found. Let me first start with animal cells. Animal cells. It is very important to know the structure of an animal cell. Now, let me just draw a membrane. Here we are going to fill out all the information, all the organelles and all the things that are inside of this cell. First of all, we have a nucleus, right? As we just discussed. This is our nucleus. And inside, we have something else, another little sphere. So this little sphere is called the nucleolus. Let me write nucleolus. Nucleolus. All right? And this is this smaller part inside of the nucleus, and it is the site of ribosomal DNA synthesis. Site of ribosomal DNA synthesis. All right, what do we have next? Now, we know that this part of the inside the nucleus, we have the genetic material. Now let me emphasize that also. Inside here we have, this is called the nucleus, and we have genetic material in this part of the nucleus. Remember, we have nucleolus inside where we have site of ribosomal DNA synthesis, and just outside we have nucleus which we have genetic material. All right. And this membrane is given a special name. It is called the nuclear envelope. Nuclear envelope. All right. Now, let us go outside of the nucleus for a bit. We have small organelles here and there. What are those? These are the ribosomes. These are very, very important because they are the machines used for protein synthesis. Let me write protein synthesizing machines. All right, these are very, very important for that reason. And then we have other organelles, a little bigger, throughout the cell. And these ones are called peroxisomes. What do these organelles do? They oxidize fatty acid. We will come what are fatty acids throughout these lectures. And then when we continue, we can start to notice something. First of all, I want to ask, how does these cells remain in a spherical position, form? Now you might be thinking, well, what else might it be? But remember, they also divide, they also have some movements, they enlarge and so on. So how do they do that? They do it by cytoskeleton, which is like a set of like wires extending across the cell, very thin layers, wires, sorry. This is the cytoskeleton. And as we just said, it supports the cell, and it aids in the movement of organelles. All right. Now let us see another organelle. This one has a similar shape, and this is lysosome. What does lysosome do? It degrades intracellular debris. It degrades. So why is degrading important? So let's say mitochondria have just used up a resource and it is floating around in the cell. Somebody needs to degrade it, right? Now, lysosome is the organelle which has this function. Let me write. Degrades intracellular debris. All right. And then, now, how can the lysosome take something, or how can the mitochondria reach out for its resources, or how can the nuclear genetic material get inside the nucleus? This is all done by a transport vehicle, which is called a transport vesicle. Like you can think of it like a little shuttle which brings all the necessary stuff to its place. So let me write it here. These are just small organelles, and these are called transport vesicle. It shuttles lipids and proteins between endoplasmic reticulum, Golgi, and plasma membrane. Shuttles between lipids and proteins. Yeah, shuttles lipids and proteins to ER, Golgi, and plasma membrane. All right, great. And then we have a Golgi complex, which is something like this. It has a weird shape. This is our Golgi complex. All right, it processes and packages and targets proteins for other organelles. It is basically a packaging organelle. Processes, packages, and targets proteins for other organelles. Okay, now we have another organelle, which is just near the nucleus. This is the endoplasmic reticulum. This one is the rough endoplasmic reticulum. And we have the smooth one, of course. What does rough endoplasmic reticulum means? It is the site of protein synthesis. Synthesis. So what was responsible with synthesizing proteins? It was ribosomes, right? So we drew the ribosomes with this color. So the ribosomes are actually on the top of the rough endoplasmic reticulum. We can just draw them attached, because they have the same function. All right, and then we have mitochondria, which is really, really important, like this. These are the mitochondria. Let me write it. Mitochondrion. All right, what does it do? It oxidizes fuels to produce ATP. It oxidizes fuel to produce ATP. All right. And lastly, but not least, we have the smooth ER. And eventually we are at the end of our organelles, and we have here a smooth endoplasmic reticulum. Okay. What does it do? It synthesizes lipids and it metabolizes drug. It is very important because it is its task is to make drug metabolism, which is really, really important. So let me write it with a little darker color so it is easier to read. We have lipid synthesis and drug metabolism, which is really, really important. All right. Now, in the next lesson, we will also cover the plant cells. See you in the next lesson.